Anyone who drank milk as a baby carries a working version of the gene that codes for lactase. In lactose-tolerant individuals, that gene keeps working into adulthood, producing the protein that digests lactose. But in people who are lactose intolerant, the lactase gene is switched off after weaning. Approximately 65% of the human population has a reduced ability to digest lactose after infancy. Eastern and North African populations domesticated cattle between 7,000 and 9,000 years ago, and these animals were later brought into Europe. The mutation spread throughout Eurasia to Great Britain, Scandinavia, the Mediterranean, India, and all points in between. Independently, other mutations for lactose tolerance arose in Africa and the Middle East, though not in the Americas, Australia, or the Far East. In an evolutionary eye blink, 80% of Europeans became milk drinkers. In some populations, the proportion is close to 100%. Though globally, lactose intolerance is the norm. Around 2/3 of humans cannot drink milk in adulthood. In that cow-friendly environment, being able to drink milk directly instead of having to process it into lower lactose cheese was advantageous, providing additional sustenance and, during droughts, a source of water. The lactose tolerance mutations arose randomly, but once it arose, it had a distinct advantage in these populations. People carrying the mutation could drink milk their entire lives. Lactose intolerance in adulthood is caused by reduced production of lactase after infancy. If individuals with lactose intolerance consume lactose-containing dairy products, they may experience abdominal pain, bloating, nausea, and diarrhea beginning 30 minutes to 2 hours later. Lactose persistence and non-persistence reflect inheritance of different alleles of the lactase gene. Lactose persistence, and therefore lactose tolerance, is inherited as a dominant trait. Lactose intolerance is the result of being homozygous for the recessive lactase allele that is poorly expressed after early childhood. Being homozygous or heterozygous for the persistence allele allows lactase expression after the time when lactase expression is normally downregulated. In some circumstances, heterozygotes can manifest partial intolerance, indicating that this is an incompletely dominant gene. Lactasia is a disorder in which infants are unable to break down lactose in breast milk or in formula. This is a rare disorder. Its incidence is unknown. This condition is most common in Finland, where it affects an estimated 1 in 60,000 newborns. This form of lactose intolerance results in severe diarrhea. If affected infants are not given a lactose-free infant formula, they may develop severe dehydration and weight loss. The frequency of lactase persistence is high in Northern European populations, decreases in frequency across Southern Europe and the Middle East, and is low in non-pastoralist Asian and African populations. Notably, lactase persistence is most common in pastoralist populations from Africa.